Revolutionizing human genomics. CU Anschutz builds the most inclusive human genome map yet. Imagine a world where genetic research truly reflects the vast diversity of humanity. That's what a groundbreaking study by an international team of scientists, including researchers from the Norman Lab at the University of Colorado Anschutz Medical Campus, CU Anschutz, has achieved. They've created one of the most comprehensive and inclusive maps of the human genome to date, published in Nature. This study involved sequencing the DNA of 65 individuals from diverse global populations, resulting in 130 genome assemblies. Each assembly offers a detailed and accurate view of an individual's genetic code, capturing the full spectrum of human genetic diversity. But here's where it gets controversial. Until now, the reference genome used by scientists was based on just a few individuals, primarily of European descent. This meant that many genetic differences, especially those found in underrepresented populations, were missing. The new Pangenome addresses this gap, providing a much broader range of human diversity. This is crucial for researchers to better understand how our genes vary and how these differences may relate to health and disease. By focusing on the most complicated parts of the human genome, such as the NHC region that houses the HLA genes, this study encompasses those areas most commonly linked to complex diseases, explained Paul Norman, Ph.D., a professor of biomedical informatics at CU Anschutz, who contributed to the study. These genes are involved in multiple immune-mediated diseases, including autoimmunity, allergy, infection, and some cancers, as well as their treatments, such as transplantation and specific immunotherapies. They also play a role in effective placentation. This resource is relevant to a wide range of clinicians and scientists on campus. Filling in the blanks, the team successfully closed 92% of the gaps found in earlier genome maps and fully completed 39% of all chromosomes from end to end. This includes some of the most complex and medically important regions of our DNA, such as the NHC region, which is crucial for immune response, genes like SMN1, SMN2, which are linked to spinal muscular atrophy, AMY1, AMY2, which aids in starch digestion. They also fully mapped 1,246 centromeres, the central parts of chromosomes essential for cell division, and discovered surprising variations in their structure. These findings could have implications for understanding certain diseases. A new era for genetic research. By combining this new data with earlier drafts of the Pangenome, scientists can now detect over 26,000 structural differences in DNA per person, many of which were previously undetectable. This level of detail allows for more precise genetic testing and a deeper understanding of how our genes affect health and disease. The study represents a significant advance in genomic research and a meaningful step toward more inclusive and precise medicine. The potential impact includes improving the accuracy of genetic testing for people of all backgrounds, enabling better diagnosis and treatment of rare and complex diseases, enhancing research into conditions like autoimmune disorders, neurological diseases, and cancer, supporting more equitable participation in precision medicine and clinical trials. Because we had developed cost-effective methods to analyze the HLA and MHC at high resolution, and through our ongoing goal, in collaboration with the Dilthey Group from Dusseldorf in Germany, to increase the diversity of genomic references, we were asked to contribute to the study, said Nicholas Pollack, a postdoctoral candidate in human medical genetics and genomics. We are now making the high-resolution genotyping methods available to anyone on campus who is interested in the role of HLA and MHC in human health and disease. This research opens the door to more precise and effective medical care that better accounts for the genetic differences across global populations. Featured experts: Paul Norman, Ph.D., Nicholas Pollack. Unlocking human genetics. 65 genomes reveal hidden secrets. Imagine this. The Human Genome Project, once hailed as the pinnacle of genetic exploration, has taken a massive leap forward. But here's where it gets controversial. The initial draft, completed in 2003, was just the tip of the iceberg. Fast forward to today, and we're diving deep into the genetic diversity of our species with the 1000 Genomes Project. But this is the part most people miss. Two teams of researchers have re-examined some of those 1000 genomes using cutting-edge techniques to correct errors and piece together the genetic puzzle more accurately. Let's dive into the details with our experts. Dr. Christine Beck from the University of Connecticut Health Center, Dr. Glenys Logsdon from the University of Pennsylvania, and Dr. Adam Philippi from the National Human Genome Research Institute. Welcome, everyone, to this eye-opening discussion. So, Dr. Logsdon, let's start with the big picture. What were you and your team aiming to achieve with this study? Dr. Logsdon, our primary goal was to generate complete sequence assemblies of 65 diverse human genomes from around the world. We focused on resolving all sequences within the 46 chromosomes of these genomes, including the most challenging regions that have baffled scientists for decades. These regions are crucial for understanding genetic and epigenetic variations, how our sequences and structures differ, and how proteins vary among us. One of the most intriguing areas we resolved are the centromeres, essential chromosomal regions found on every chromosome. They ensure that chromosomes are accurately segregated during cell division. For the first time, we solved about 1,200 centromeres from these 65 genomes, revealing how they differ and what those differences mean functionally. Ira, that sounds fascinating. Does this mean the older genome sequences had significant issues? Dr. Logsdon, absolutely. Sequencing technologies from about a decade ago couldn't resolve these complex regions due to their repetitive and large nature. The data generated back then was smaller than the size of the repeats themselves. But now, with advanced techniques, we can traverse these regions from one side of the chromosome to the other, providing complete, high-resolution maps. Ira, Dr. Beck, tell us more about the 65 individuals whose samples were used. Where did these samples come from? Dr. Beck, these 65 samples were part of the 1000 Genomes Project, collected from diverse populations worldwide. We chose cell lines from individuals that would maximize the discovery of novel sequence variations. If we sequenced similar individuals, we'd get less valuable data. By sequencing these 65 people, we uncovered a vast amount of DNA variation. Ira, so you found a lot of diversity and dissimilarity in these genomes? Dr. Beck, exactly. We cataloged over 188,000 variations between people that were greater than 50 base pairs in length. This isn't surprising given the complexity of these regions. Previous studies had limitations, but with our advanced techniques, we uncovered differences that were previously undiscovered. Ira, Dr. Philippi, what technological advancements are enabling this work? Dr. Beck. Philippi, it's a combination of better machinery, improved tools, and more powerful computers. The 1000 Genomes Project, started nearly 20 years ago, had significant gaps due to repetitive sequences. These repeats are like missing pages in a book, hard to piece together. But now, with longer read sequencing technologies and advanced computational methods, we can fill in those gaps and assemble the genome more accurately. Ira, how complete do you think these genomes are now? Dr. Philippi, we estimate that these genomes have about 8% more sequence than the initial Human Genome Project from 2003. The technology allows us to read longer stretches of DNA and assemble them more accurately. Handling the sheer quantity of data is only possible with the advancements in computing over the past couple of decades. Ira, are we close to a final, complete human genome sequence? Dr. Philippi, not quite. We're just scratching the surface. But by sequencing the most genetically diverse individuals, we maximize our return on investment. In the next few years, we'll be sequencing thousands of genomes, and the sky's the limit with exponential advancements in this field. Ira, Dr. Logston, what does the repetitive nature of these genomic regions tell us? Why do they repeat? Dr. Logston, these repetitive regions are not junk DNA. They are functional and essential for life. For example, centromeres are highly dynamic and variable. Variations in these regions can affect chromosome segregation during cell division. Understanding these repetitive regions is key to comprehending genetic diversity and function. Dr. Beck, absolutely. These repeats, whether centromeric or segmental duplications, provide insights into genetic variations and their potential impacts on gene function and regulation. Getting a clear picture of these repetitive sequences is vital for understanding genetic differences and their downstream effects. Ira, Dr. Philippi, how much data do we need to determine what is normal genetically? Dr. Philippi, it's a great question. We're building a foundation of what typical human genomes look like. In the next few years, we'll see genomic language models trained on this data, making accurate predictions about genetic variations and their disease associations. The more data we have, the better we can predict the effects of genetic mutations. Ira, what kinds of predictions are we talking about? Dr. Philippi, imagine mutating a random base in your genome. With our advancing technologies, we're getting better at predicting whether that mutation will be deleterious. We're focusing on non-coding regions, where most mutations are benign, but the few that matter are crucial. We're on the brink of making these predictions accurately at birth with DNA sequencing. Ira, Dr. Beck, how has the Human Genome Project fared in its goal of curing illnesses? Dr. Beck, the sequencing of the human genome has been a game changer for Mendelian diseases, where there's a clear cause and effect relationship. However, for complex diseases, we're still unraveling the genetic architecture. Machine learning and a better understanding of genetic variation will be key to making progress in this area. Dr. Philippi, the initial Human Genome Project was a monumental investment that has paid off immensely. The cost of sequencing a human genome has dropped dramatically, and the economic and health benefits are enormous. This project is a gift that keeps giving. Ira, thank you all for this enlightening discussion. We look forward to more updates on your groundbreaking work. Please join us again soon. End of transcript.